Okay, hi everyone, and welcome to this next session. And hopefully we have all the people coming in that are trying to make it in as fast as possible. So it's my great pleasure to chair this next session. My name is Pasi, those of you who didn't catch that yet, from Turku Bioimaging. And the topic of the session is data management and ethics, but we will also have a little bit of a special things included in this session, so also a talk from Bioimaging, and then at the end, so don't escape, because then we will have all the prizes as well for the picture competition and the best poster. And then after that, we will be taking our group picture. So information about that at the end of the session. Mm-hmm. But to kick things off, it's my great pleasure to introduce Jason Swedlow, who I think doesn't really need much of an introduction, but the founder and head of OME. So Jason, please go ahead.
That's great. Thank you, Pasi. So it's courteous and professional to say thank you to the organizers for the invitation. So thank you to Pasi for the invitation. But seriously, thank you so much. It's great, so great to be back at OME. It's great to be with all of you. And so it's been a great couple of days. So I'm going to tell you about what we've been doing in OME for, close on 22 years now. But I'll give you an update of recent work. I'll mention some of the stuff that's going on in Glencoe software. And then also, if I have time at the end, kind of a bit of a departure, but I think there's a few things that might be interesting. Work that I've been leading at Wellcome Leap. Previously, I've been associated with Bioimaging UK and Bioimaging. You'll hear about Bioimaging later from Anta. I've stepped back from those roles. Okay, so first of all, the commercial disclosure. So everything I'm going to tell you about from OME is completely open source. It's available. By about 2005, 2006, we were having some success, and we had biotech, pharma companies, technology companies coming to us saying, form a company. We want access to this, and we want ways to do that, to access this in a commercial context. So with basically not much more than that, we formed Glencoe software. And so that's what Glencoe is about, trying to find other ways to disseminate this work, in particular to biotech and pharma. So that's where that's coming from. Okay, so the problem, imaging data is everywhere. There's been lots of discussions here. And we discuss this every year at ELMI, and the data sets are larger and larger. Obviously, we all have the privilege of being imaging scientists, so we have great stuff to put in our PowerPoints, and those are pretty pictures. The reason we do all of this is the measurements that we make, and I won't belabor that point in this audience. But really what we've been working on in OME is this idea of making these data sets a resource. And so that's a resource for collaboration, a resource for exchange, a resource for validation, a resource for publication. And so we mean this word resource in the kind of broadest possible way. That's really what I'll be talking about today. Okay, so typically in OME, we don't do any of the interesting stuff. So you have great speakers this morning, Anna, Ricardo, and others talking about really amazing analytic technologies. You've seen lots of amazing imaging technologies presented here. So lots of cool microscopes, lots of cool analytics. That's great. That's not what we do, okay? We're the stuff, we try to be the stuff in the middle, and the idea is to be able to connect these different parts, the sort of the peripheral parts of this diagram, and make them work together. And make them work together in a way that's seamless, and so that a user wouldn't know that, in fact, something was going on underneath to manage that exchange of information. So there's lots of words for that. Interoperability often comes up, central broker, APIs. There's lots of words that kind of are associated with this idea, and I'll mention a few of them as I go on. But it's all about trying to understand what the data is that's coming in and what it needs to actually be, how that data needs to be communicated. That's effectively what we do. Okay. Now, as you'll see, we've been doing a little bit more in visualization, a little bit more in analytics as we go on, but our core is right here in the middle. Okay, so brief words about the technology. Again, all open source, all freely available. These are words that you can find on Google or whatever you choose to search on. I want to emphasize that we start out with a metadata specification. It's called the OME data model. It's nothing more than a bunch of text that talks about different metadata types. I have to confess that when we started this out early 2000, 2002, we were very conscious not to go anything farther than a fluorescence microscope. We didn't handle the experiment. It was too hard. We didn't know what to do. Others have taken on that challenge. The work in Quarup that has been so powerful recently is really, taking on those ideas. And so what's been great to see is that that core model that we came up with nearly 2 decades ago has been at least helpful for others to extend from. And so we've been working with Caterina Strambro de Castilla and others on this. And so I think that it's a great example of how the community can leverage each other's work. Our implementations of that model are sort of below here. So we have a file format, OME-TIFF. I won't talk too much about that, but I'm happy to talk about that. I'll talk about a bit about bioformats in Amero. Spend most of the time on IDR and a little bit on our most recent work, this a new file format that we hope is important, will be useful for the future. Okay, so quickly, bioformats. Many of you use this. It's in Fiji, it's everywhere. It is nothing more than a proprietary file format translator. Project was started in collaboration with Kevin Elsery's team at Madison. Lots of proprietary file formats, lots of ways of writing down the binary data, the metadata. And so what is an experimentalist or what is a user to do when you've got hundreds of different file formats? And so bioformats has readers. We reverse engineer the different file formats. We install readers inside of bioformats, and then anything that uses bioformats, and there's a lot of open source as well as commercial software that uses bioformats, now becomes able to address effectively that panoply of data types. Okay? It's built on contributions from the community, so thank you all for sending us data. By our experiences is that the scientists are the best source of sampling the data, and it's used heavily all over the world, and that's great. So bioformats has been certainly one of our success stories. I'm going to tell you, however, about later on in the talk about how bioformats is becoming limiting, and we have to think about alternative ways of working. Amero is our full-blown data management system. So those of you with a computational background will recognize sort of a 3-layer structure here. So data sources on the bottom, middleware application. This is a server connecting through an internet application, standard secure internet, to clients, so a server-client architecture. These clients can be in any of the frameworks that you would normally use in a computational environment, lots of different programming languages, web, et cetera. So the idea is that this provides a common interface to access, for example, the imaging data, the metadata, any associated analytic data. And the idea here is just that we're doing what everyone else is doing. This is how you read your email. This is how you read your newspaper. You effectively have a client. Connect to a server. Yeah, there we go. Client connect to a server. That server amalgamates data, sends information back to the client. So we're just trying to do the same thing that's common in the rest of the world to do that with bioimaging data. And so this is how we sort of construct systems. So in any of your facilities, lots of different data types coming off of various imaging systems onto large file stores, probably multiple of them. So bioformats is your single point of access to those many different file formats. And then we put Amero on top of bioformats. Lots of people put other things on top of bioformats. We put Amero, and Amero becomes your single point of access to effectively everything underneath here. And so I'm going to show you lots of screenshots here. Most of these will be web client screenshots, so that effectively this is me on my laptop connecting into a Amero server, going through bioformats, pulling data. So those screenshots, you're going to have to imagine that everything, all of this stuff is underneath is here. I'm going to show you a screenshot here, but the rest of the system is working. Now, the other thing we can do is connect into lots of analytics. We've done that. Lots of other people have done that as well. This is all published. See the website for more information. If you look up Amero, read the docs, I think, you'll find just a huge number of documents set up by the team for how you do these integrations and the details of how making this all work, both open source and commercial tools. Okay? So I won't say much more about analytics in this talk. Other applications that we've built, this is Amero Figure. A lot of people have called this the killer app. So now, traditionally a figure in a paper are screenshots of data, for example, in well, maybe Adobe Photoshop, but you take a screenshot and then you drop things into Illustrator, whatever, and you make it this nice figure, you put A, B, C, etc., and you send that off to the journal. And then hopefully not too many weeks later, you get the response back, and now you've got to go and sort of alter those figures. And the challenge has always been that, oh my gosh, where was that data set that I put into figure 3A? I have no idea what that was. So these are not screenshots. These are actually calls to the database. So this is an application running on top of the database, and the figure is all assembled from calls from the database. So this means that effectively the provenance of the data that's sitting in a figure is maintained. It can be shared and all that kind of stuff. So this has been very popular. More information, just Google Amaro figure. Okay. So switching over, so I wanted to talk a little bit about some of the things that Glencoe is doing, because I think it is relevant to this audience. And so there's lots of things that are challenges in this space, in this data space. But one of the things that people talk about is scale. Okay, so now I want to be working at the petabyte scale. I actually want thousands of people accessing very, very large data sets. How does that work? So this is sort of another drawing of environment, so-called classical Amaro deployment. So here's your back-end storage. Here's Amaro server. Remember, I mentioned a web client. So here's a screenshot of a web client. Amaro's talking to a database. In this case, it's a database called Postgresql, but it doesn't really matter. And this is how most Amaro's sort of out-of-the-box or vanilla-flavored Amaro would be set up. And this is fine, and it works quite well. But note that there's sort of a single point of access here. And if there's lots of requests or lots of data, eventually that single point of access gets saturated and things start slowing down. That's just natural. So what do? Well, you start stacking up more servers. Okay. So this is called horizontal scaling. So what Glencoe has been doing is making so-called microservices. So mini Amaros, so Amaros that do just a little bit of what Amaro does, but don't do it really well. And so, for example, looking at image regions or handling pixels or doing thumbnailing. This turns out to be really important in high-content screening where you have to calculate a ton of thumbnails. And so this idea has been very important, for example, in drug discovery, but also, as you see at the top of the screenshot here, and I'll just blow this up, now in multiplexed imaging, this sort of 40, 50 channel imaging of tissues, very, very large images, hundreds of pixels in dimension, many, many different channels, and making this all work. And so this screenshot is data from Peter Sorger at Harvard Med School, but this is Glencoe's built an application called PathViewer that effectively leverages all that. If you want more information, check the website. Okay, so that hopefully gives you an idea of how both Glencoe and OME are working together on these problems. Okay. Last few minutes, let's talk about data sharing and publication. So most of you will know in the structural biology, for example, in genomics, absolutely routine to publish the data, for example, a genomic sequence or a structural, a 3D structure file of a macromolecule we're working with. How do we make that happen in imaging? And so what you're seeing here are screenshots, again, of various clients. This is the Empire resource at EBI. This is Amaro underneath serving images. So this is this server-client idea here at work. So lots of different types of clients can attach to an Amaro server. This is at Riken. This is our colleague Shuichi Onami. Ken Ho is here, who used to work with Shuichi. So Ken did a lot of this work. Again, there's a Amaro sitting underneath here with different clients, and effectively Amaro's handling the metadata and the imaging data. Okay, so I've cut a lot of this short, but there's now a lot of examples of people using Amaro to publish data, for example, for their project or from the institution. We hear about this acronym FAIR. There are words here, and they actually have definitions, and Amaro can do all of these and make all of these happen. If you want more information, we published blogs on this, and you can reference the original paper. So we saw this emergence of many different institutional repositories, and that made us start thinking, okay, what about basically a community repository for all of the imaging data? And that started us down this road of building what we call the Image Data Resource, or IDR. And so this is a project that we did in collaboration with EBI, with Eurobioimaging, many others. The IDR is up and running at the European Bioinformatics Institute in Kingston. They have a collaborative cloud called Embassy, and Everything's running there. There's the URL. So you may recognize this sort of layout of this UI. This is our web client, Amaro Web. So it's an Amaro, but a finely tuned Amaro with lots of different types of data, lots of annotations and analytics, download, et cetera, all bolted onto us and running at scale. So just a quick statistics. This is a little out of date, but gives you an idea of the scale that we're working on here. Several hundred terabytes, lots of imaging data, and so on. But critically, for what I'm going to talk about here, lots of different annotations, so lots of extra information associated with the images. It's heavily used, about 78,000 unique IPs in 2021, 34 million hits, so thousands of people hit IDR every day. And what we're doing, so this is not data from my lab, I hope that's clear. What we're doing is we're publishing data sets that are associated with publications coming out in various journals. And so in the data availability section then, so just like in the structural biologist would have a PDB entry, we would have effectively an ID or an accession number for data sitting in IDR, okay? Now I'm gonna highlight here, note the number of data sets we published. It's closing in on 130 now. And you might think, oh, that's actually not a high number, and just hold that thought, because I'll get back to that. But we've been doing this now for several years, since 2016. It's working fairly well. Lots of different data types, light sheet data. This is from Philip Keller's lab. This is Katie McDole's definitive study of the mouse embryo fate map. Human cardiac biopsies. This is a machine learning study from Anat Matabushi's lab. And the original imaging data from the tissue atlas, part of the human protein atlas, the JPEGs were on their website, but they wanted to publish the data. And so we've been working on them, and I think we have like 11,000, 12,000, something like that of their antibodies now published, and we're working to continue that. Just one example of what we do with the different metadata. So this is a data set from Fei Chen's lab in the United States, and this is in situ genome sequencing, so thinking of new ways of thinking about identifying locations of genomic regions. And so we have the imaging data, which is fine, and note we have lots of metadata here, which I won't really go into, but all of that's there. But the key point is we have all of the regions, all of the spots we have located, and for every spot, we have effectively, obviously, its position, but all of the sequencing data, the UMI information, and so on, to effectively define what that spot is. Okay, and we thought that was a good idea. And that's all we did. And we thought, okay, hopefully that's useful. And then Jean Ma's lab at Carnegie Mellon came along and built a public nuclear structure viewer. And so they took that data. So they have the URL is here, the bioRxiv paper is here, and they've now basically taken that data and making viewers. And so if you want to find out where, for example, chromosome 6 is sitting in this case, human and embryonic nuclei, you can do this. We've done this now with several other studies. And so initially, we just did this as a sort of one-off because that was all we had access to. Around the time that we published this study, Katarina's work at the 4D Nucleome project in the United States was defining a specification for how this metadata would be written down. We've now adopted that, and so we're continuing to drive that forward. Hopefully I get another example of how these different projects are learning from each other and moving forward. So I mentioned lots of metadata. I don't have time to tell you a lot of the details here, but we employ a whole series of different ontologies to describe, for example, the experimental details all the way down to the details of the chemistry, for example, for drug screens, et cetera. And all of that is searchable and so on. Okay? Now, this issue, I said we've only, so since 2016, we've only published on the order of 120 or 130 studies. It's not very many if you think about it. And so what's going on? And the key point is what IDR is focused on is so-called reference data. Okay, so these are data sets from many different domains. So we're sort of technology and domain agnostic. But what we're looking for are comprehensive studies across, for example, genes, chemical compounds, cell lines, biological states, et cetera, where the data end up, for example, being some sort of story, and then all of the curation that I've just been showing you would really add significant value to the data. So a so-called added value resource, okay? That's what IDR is, okay? It is not, and if you think that through, most of the data sets that are associated with publications don't fit that criteria. Okay, so we're not going to publish the vast majority of the imaging data that's coming through all of you and the rest of the community. And so this idea of reference images came out of a collaboration with Eurobioimaging and Elixir, the European life sciences informatics infrastructure, working with Antje and other colleagues. And so all we did was take that idea and then implement it. But that raises the question of, what about everything else? And so here, I have to tip my hat to Matt Hartley, who's taken on the job of building the Bioimage Archive. And Matt, your poster is at, what's your poster number? Yeah, okay, it's in the side room. Go see Matt's poster. It's really important to understand what's happening here. So Matt's building the resource that's going to hold, by and large, most of everything. IDR, we're focused on these very sort of cell-based and tissue-based data sets, these reference data sets. And we're working quite closely with Matt's team to work out the details of metadata, et cetera. So this idea was sort of written up a few years ago in Nature Methods. And so since then, we've been working on implementing all of this. And don't pretend that this is a 2 or 3 year project. This is a long-term labor of love. Matt's smiling. I think he's smiling or crying. But anyway, the key is that these ideas of separating these different types of functionality are things that we're adopting that have been learned in other fields, genomics and structural biology. This is the layout for imaging. It's not just, for example, here in Europe. Shuichi Anami and colleagues have been doing this in Japan as well. The US is finally coming online and starting to think about this as well. So this is really a global effort now that's building up. That's very exciting to see. Okay, so I mentioned bioformats. And if you think about it, real success story. So lots of different proprietary file formats coming from the various vendors that make amazing systems, and they're writing different formats. That's actually necessary, so don't ding this. This is actually important. Bioformats, as I said, is sort of the common way of accessing that data with various translators. And so bioformats then plugs into lots of different tools, commercial tools, open source tools, and basically gives them access to all the metadata, the binary data, et cetera. Not technically very interesting, but in terms of data access, very important. One of the problems we've seen is there's more and more vendors, and there's more and more formats, and this just keeps going on. And if you think about this diagram in the context of what I just showed you, IDR, you might start thinking, there's kind of a problem here. There's a scaling problem here, because this single point of sort of translation is going to start being a bottleneck. And I can tell you, in IDR, bioformats is a massive bottleneck. Okay? The need to identify all of these different data types, et cetera, is causing us real pain. Okay? So what have we been doing about that for the last several years? Josh Moore, who's here, has been leading this effort, trying to come up with new file formats that would be scalable, that would work in the cloud. So you may have heard this acronym OME-NGFF. I won't go into technical details. This is all published in Nature Methods in December. There's a specification online. There's open source software, again, from the open source and the commercial community, trying to take these efforts forward. And so this will be a long-term effort. It won't be an out-of-the-box solution that just appears in 2 or 3 weeks. This is multiple years in the making, but it is definitely happening. And so what that will mean is that most of bioinformatics and most of the imaging world is in this using data download. There are now applications. So this is Napari. There's a Python application, but now there are plugins. The Moby plugin from Christian Tischer at EMBL. There's just the bioarchive paper. Now actually read these scalable formats and make our desktop clients able to access this data at scale. I'm deliberately avoiding all of the technical details because Pazi's standing up already, and actually this is all published, so see the paper. There's online examples, so Google IDR NGF samples, you'll get this, and you can see this stuff working. And so this idea of, oh, my light sheet data set is so large, nobody can access it, maybe that was true a couple of years ago, but it's not true now. Okay, you can share these data at scale, and you can transmit these data, and you can feed them into machine learning applications. So what that means is that those of you working on the desktop, proprietary file formats on one side, you've been depending on bioformats to be your translator into your analytics. that works. I talked about a Marrow, some sort of server application, but we still have that translation. And key, and I apologize for not emphasizing this idea of on-the-fly translation. So the data is stored in these proprietary file formats and then translated as access. And it's that on-the-fly translation that's so important. Now what we want to do is to convert one time and put these data into a format that can be streamed. And that's really important. You don't anymore go to a video rental shop and take home a piece of media with the whole video on it and play it in your house, okay? You don't download the whole data set and bring it with you on your laptop. What you do is you stream data, and that's what we need to do in bioimaging, okay? That's what this is all about, okay? Now, if you think about that, where's that going? So this is notional. This does not exist. Please don't start asking me about if this is going to be available next Tuesday, because it's not. But this is what we will be working on. Data sets now are getting so large, hundreds of terabytes of data sets coming out of several institutions worldwide, that they will be written once onto commercial clouds or perhaps institutional clouds, and there they will stay for the rest of eternity. And so IDRs, archives, et cetera, are going to have to find a way to have distributed access to these data sets, and the NGFF technology allows that. Basically, this makes that happen out of the box, okay? And that's really important. This provides us a future for the types of data we're talking about. Okay, let me just, I know Pazzi is about ready to kick me in the knees, but I just want to make one very important point. So I've been telling you about this OME NGFF or ZAR format. I haven't told you about any of the technology. Josh is here. You can find about that. Please don't think that what we're saying is, oh, we've got a new format and you should use this for everything. That's simply not true, okay? Again, this was all published and we talked about this, and Josh did an amazing job of benchmarking lots of different file formats. But it turns out that different file formats work best in different conditions. Not a terrific surprise. If you're working locally on your high-powered workstation, probably you're going to be working with HDF5. If you're working in the cloud or sharing data in a repository, you'll probably be working in ZAR, okay? So we'll have to think about exactly what we're trying to do, and we're going to have to be a little bit more thoughtful about our approaches. Okay. All of this has been in a public discussion. Josh is leading this. You should talk to him about that. Just the summaries. Told you about the file formats, bioformats in a Marrow, IDR. NGFF, we think, is going to allow a lot of the technologies and ways of working that are going to be so important. And most importantly about this is that what we need to build, what we've been building and what needs to continue is this global community of all of these different entities working together. And finally, just a huge thank you to, first of all, the funding, but obviously everybody who's been involved in everything that you've seen. This is a huge amount of work, and it's been a great team to work with. Pazzi, I'm done.
Thank you. Thanks a lot, Jason. I've been following your work ever since I was a student more than 20 years ago, from the very beginning, and it's been great to become friends and work together after that. I'm afraid in the interest of time, we have to move on. If you have questions, and I'm sure you have, so please put them in the online platform into the chat, and then hopefully Jason can also there, and the others in the team pick them up later on. But once again, thanks, Jason.